All right, class, let's finish up our Chapter 12 discussion on vaccination and immunity. There are two broad categories of immunity, natural and artificial immunity. Most people have both present in their bodies. Natural immunity occurs when we are exposed to a biological pathogen through the normal course of events of our life. So we're just living our lives and we get sick randomly from an experience that we have. Maybe someone sneezes on us and gets us sick, so on and so forth. Artificial immunity is going to be obtained through some kind of medical intervention, the most common of which is a vaccine. Occasionally we'll also have immune serum administered, but that's going to not be as common. So let's focus on active immunity. Our active immunity can be either natural or artificial. Active immunity occurs when our individual receives an immune stimulus. This immune stimulus is then going to initiate the B and T cell differentiation cascade, and we're going to start making our own antibodies. We will then have memory T and memory B cells, and those memory T and memory B cells allow us to quickly respond to re-exposure to the same antigen. So that secondary response to the same antigen will be much stronger than the initial primary exposure, primary response to the antigen. Active immunity requires several days to develop. Typically, it's going to take about a week to develop. And this active immunity can last for a relatively long time, years, decades to a lifetime, depending on how it was developed. And we can have active immunity develop from either natural or artificial methods. Passive immunity, on the other hand, is going to be the injection of antibodies into a human being. So instead of exposing somebody to the antigen and having that individual make their own antibodies, instead we'll just skip the T cells and B cells and go directly to injecting antibodies into their body. Somebody is going to be, an individual that receives an injection of antibodies will be protected from the antigen for a short period of time while the antibodies are active or in their bloodstream. But these antibodies have a shelf life. They do not last forever. The antibodies don't trigger the development of memory B or memory T cells, so there's no long-term immunity with passive immunity. And if we don't have any memory B or memory T cells or any plasma cells from B cell differentiation, we also are not going to make any antibodies when we have passive immunity. The plus side of passive immunity is it's instantaneous. We instantly will get protection the moment those antibodies are injected into our bloodstream. It's very effective in the short term, and we can have either natural or artificial origins, so we can harvest these antibodies from the organisms as a whole, or we can trigger the production in a lab and purify them in a lab setting, an artificial setting. The main benefit of passive immunity is going to be antivenom applications, or if a woman has a negative blood type and we want to make sure that she doesn't destroy or kill her positively blood type child while the child is in utero. So if we look at natural active immunity, this is your classic, I got sick, I had to suffer through the illness, but I'm better for it. Now I can make my own antibodies. We also have natural passive immunity. This is, hey, I got some antibodies from mom because I sucked her breast milk, or maybe I ate their mucus, which hopefully you don't eat other people's mucus to get passive immunity, but technically it could work. Be kind of nasty, but it could work. We also have active artificial. Active artificial is vaccination. These two, active natural and active artificial, are the two most common. We also have artificial passive. This is going to be the injection of antibodies into somebody intravenously. The first event of immunizations are recorded early on in Chinese history, 6th century China. And it's kind of gross, in my opinion. They took crusts, or these healers took the scabs, the crusts from smallpox sores, ground them up, and then blew them into other people's noses. In effect, they were exposing people to a weakened virus vaccine. And they had limited success. Variolation was also developed in the 10th century, And with this process of variolation, we took the dried pus from smallpox pustules and put them into the arm of a healthy patient. So as opposed to grinding up the pustules and blowing them up somebody's nose, we would take the pustule, make a slit in the person's arm, and put the pustule in it. Variolation has been used in the Far East and was brought to England in the 1700s. The downside of variolation is the pathogen was not very weakened. So there was many live or active pathogens being injected into or being put in the patient. So many of the patients were going to still die from smallpox because they were actually being exposed to the active virus itself. In 1796, Edward Jenner was the individual who came up with the first effective vaccine. He found that dairy maids that were attacked by cowpox viruses became immune to the smallpox virus. And he tested this theory because he thought to himself, hey, maybe it's the cowpox virus that's making them immune to the smallpox virus. So he found a young boy and injected that young boy with cowpox lesions. And then that young boy developed cowpox. And 2 months later, he took that same young boy and gave him the smallpox virus. It's highly unethical, and you absolutely cannot do research like that now with all the ethical boards that are in place, thank goodness. He found, though, that the young boy, thankfully, did not contract smallpox. His earlier inoculation with cowpox rendered him immune to smallpox. This is the very first use of vaccine, and it was initially regarded with a lot of skepticism, but later was adopted when it was proven to be successful. Vaccination is a term that means giving immunity through the inoculation with selected antigens. So we're going to have passive and active immunization. During passive immunization, we try to take antibodies from other sources and inject them into human beings. The first attempts of passive immunization involved taking horse serum and injecting them into human patients to prevent tetanus and to treat diphtheria infections. It was found to have very limited success. Antisera, or antibodies from animal serum, has now been replaced with antibodies from human products. This is one of the big reasons why you should donate blood and or blood plasma, or to genetically engineered products, where we pre-program cell lines to secrete high numbers of specific antibodies. Gamma globulin is an immunoglobulin that's been extracted from pooled blood of many human donors. This gamma globulin can be used to treat very specific infections for high-risk neonates, or in other words, newborn children, or other immunocompromised patients. We use gamma globulin when there's no effective treatment for somebody who has an immunodeficiency. So anybody who's immunocompromised or hasn't had time to develop active immunity can be injected with a pooled resource of immunoglobulins from a patient, many different patients, known as gamma globulins. The basic principles behind vaccination are such. We first need to expose the patient to the antigen to initiate the primary response. And then afterwards, we're going to have a memory response. Memory cells will develop, memory B and memory T cells. We've primed the immune system now so that if they're ever exposed to the pathogen in the future, they're able to respond very quickly because they have those memory cells. If the pathogen enters the body then in the future, the response is going to be very quick, very immediate, powerful, and sustained. That secondary response is going to be much more robust. We've found that vaccines have profoundly reduced the prevalence of common diseases that used to be the big killers in the developed world. Now the big killers in the developed world are lifestyle diseases, cardiovascular, lung cancer, diabetes, diseases that are more strongly influenced by lifestyle choices than being infected with the pathogen. But 100 years ago, more people died from pathogens than they did from lifestyle diseases. What do we want to find in an effective vaccine? We want effective vaccines to expose the individual to an antigen that protects them against the pathogen they'll find in the wild. So it needs to mimic the wild form of the pathogen, whatever that pathogenic microorganism may be. A good vaccine also is going to have very few side effects. It's not going to cause bad things to the person. A good vaccine is going to stimulate both memory B and memory T cell formation. So we're going to have B cell and T cell activation. Good antibodies are going to last a long time. Good antibodies are not going to require repeated doses. The best antibodies require only one dose. And then, of course, a good antibody is going to be inexpensive and easy to give and easy to store. So, what kind of antibodies do we use? We can use whole cell or viral vaccines, and of those we can have live, living cells or living viruses being administered, or we can have killed or inactivated viruses administered. We can also just give the antigenic molecules from the bacterial or virus cells. So we can purify out the specific antigens that are specific to the pathogen and give the patient part of the microorganism. Those subunits can be derived from cultures of genetically modified cells or viruses. They can be synthesized to mimic the natural molecules. These subunits can be manufactured with genetic engineering. Sometimes you'll find a conjugated vaccine. Conjugated vaccines are going to have subunits conjugated with a protein to make them more immunogenic. Remember, proteins are larger molecules that can trigger very robust immune system responses. And if we can take that subunit and bind it to a larger protein, it'll be more likely to be caught by the net the immune system casts. So if we looked at a killed or inactivated viral vaccine, we could take our virus, we could take our cell and kill it, and it looks almost the same to our immune system, but now it's no longer metabolically active, and we can administer it to our patient, and then we can develop the immune response. Or we could take a living, fully living cell and administer it to the patient and trigger the immune response. The downside, though, is these microbes can multiply within the patient and potentially make the patient sick. So most people, generally speaking, try to stay away from or frown upon living cell or unattenuated vaccines. A big advantage, though, of the living vaccines, though, is that they are more effective, they don't require booster shots, and they have a longer shelf life. And they provide more, they provide a longer lasting immunogenic effect. So you're immune to the pathogen for a longer period of time. The big downside, though, is you may potentially get sick. We also could use subunit vaccines. Subunit vaccines are going to be individual molecules that are isolated from the virus, the bacteria in question. Oftentimes, this individual molecule can be produced from a genetically engineered microorganism that then secretes the individual protein or toxin that we can then administer to the patient. Right now, many vaccines are currently in the pipeline. There are many different bacterial, viral, and protozoal and fungal diseases that don't have functional vaccines. Malaria and HIV/AIDS are two of the biggies right now that we're trying to develop vaccines for. There are many other diarrheal, respiratory, and other worm, helminth-based diseases that can affect many different people every year. Approximately 200 million people every year are affected by these diseases. Even the vaccines that we do have aren't available to many of the people in the world. And these difficult to design vaccines for latent or persistent viral infections are quite difficult to get rid of. Remember, latent means that the virus can hide in the host cell genome. So if we look at herpes virus or cyclometaviruses, these viruses can hide their genome in the nucleus of the host cell, and it makes it very difficult to develop a vaccine for these viruses. The host's natural immunity can't clear the virus, so the artificial immunity that we inject or expose or try to induce in the host needs to outperform the host's natural response. And that's a tough sell. It's tough to make the immune system be more effective than what it normally is. Some vaccines are DNA-based. We can take microbial DNA, put it into a plasmid vector, and then take that plasmid vector and inject it into the patient. Our human cells then will uptake that plasmid vector, and start to make some of the microbial proteins in our own bodies with our own cells. These foreign proteins then can then be recognized by our immune system, and our B and T cells can then be sensitized to that specific protein that's from that specific microbe. These DNA-based vaccines have been shown to be safe in animal studies, and we can design them to emit or secrete any specific microbial protein. Our favorite protein can have its gene inserted into the plasmid and then be used to make a DNA-based vaccine. So to make a DNA-based vaccine, first we need to take the gene in question, remove it from the pathogen, put it in a plasmid, inject the plasmid into a human. That plasmid then goes through the bloodstream, is sucked up into the host cell, and then is incorporated into the nucleus, and then the host cell starts to make the new protein that's associated with the pathogen and can trigger the immune response. So you now are going to be immune to the initial microbe that protein was unique to. There are some side effects of vaccines that are worth talking about. Vaccines, generally speaking, are going to be administered subcutaneously, intramuscularly, or intradermally. So they're going to be injected into the superficial body fat, they're going to be injected into a muscle, or they're going to be injected into the skin on the surface. We also can administer vaccines by snorting them up the nose or by eating them. Nasal and oral vaccines, though, are the exception. They're only available for a few diseases. These nasal/oral vaccines are going to focus on stimulating IgA production in our mucous membranes. The advantage of these vaccines is that they're easier to give than the injectable vaccines. So for anybody working with toddlers, you know exactly what I'm talking about. And they're very rarely accepted and well tolerated. There's very few side effects with these nasal/oral vaccines. To help our vaccines be more effective, we'll oftentimes mix them with a chemical that gives us a more robust immune response. These chemicals are called adjuvants. They enhance the immunogenicity of our vaccines and make it so that the antigen can stay at the injection site for a longer period of time, and makes it so that it takes a longer period of time for our body to process and remove the antigen. And by keeping that antigen at the injection site for a longer period of time, we can maximize the exposure to our leukocytes, or our white blood cells. The most common adjuvant used currently is aluminum hydroxide salts, aka alum, which is also used to make excellent meringues for those of you who are interested in making a meringue or a banana cream pie. Now, vaccines need to go through many years of clinical trials and experiments in both animals and human volunteers before they're given to the public for general use. Now, even though they go through this extensive process, there's still going to be some complications. Sometimes there'll be a local reaction to the injection site. Having a little bit of swelling and redness at the injection site is a rather common vaccine. We also could potentially have a low-grade fever or a low-grade allergic reaction. Every now and again, there'll be more adverse reactions, but these are statistical anomalies. Rare reactions can include panencephalitis to the measles vaccine. Panencephalitis is the swelling or inflammation of the entire brain. Itis meaning inflammation, encephal meaning brain, pan meaning the whole one, or the entire. We could also have a back mutation to a virulent strain. So if we were to administer a non-virulent strain of the active microorganism, that non-virulent strain can become virulent after being administered. We occasionally are going to have diseases caused by vaccines because the vaccine was contaminated with another pathogen or a different pathogen. Sometimes there can be a neurological effect that's associated with the vaccine, but doesn't necessarily have a specific cause that's known. And this has been found with the pertussis and swine flu vaccines, but again, these are very rare reactions. And then finally, sometimes individuals are actually going to be allergic to the medium that that vaccine is contained in. For example, some vaccines are going to be made using chicken embryos. And those vaccines will have egg components in them that some individuals are going to be quite allergic to. It's worth mentioning that there's no established link between autism and the mumps, measles, and rubella, the MMR vaccine. Initially, in 1998, there was a paper published linking autism to mumps, measles, and rubella. It didn't say that it caused it, it just said that there was a correlation, not causation, between this vaccine and autism. However, everyone involved with the study eventually disavowed it. The journal article, the journal, the Lancet that published the study, revoked the study, and the primary author had his medical license revoked. Since then, there's been multiple studies done involving tens of millions of data points, and it's found that there's been no link. This initial study had only 12 data points in it. And then if we combine all the data points from the other studies, we have over 10 million data points contradicting the initial 12. In 2011, the Institute of Medicine stated unequivocally that the mumps, measles, and rubella vaccine is not causing autism. Unfortunately, though, that paper that was published back in 1998 caused a lot of people to become very wary and very scared of getting vaccinated. And the rate of vaccination in developing countries now has been dropping off. Many parents are choosing not to get their children vaccinated because they're worried about some potentially bad side effects of vaccines. Many parents believe still that vaccines cause autism. And because now people are oftentimes choosing not to get vaccinated, there's been outbreaks of mumps, measles, diphtheria, polio, typhoid fever, and whooping cough, diseases that shouldn't be in developing countries and that were nearly eliminated or formerly eliminated in developing countries. One of the reasons for this increase is the decrease in herd immunity. If almost everyone in the population is immune to the disease, it's very difficult for the disease to pass through the population and get to the individuals that are actually susceptible to it. For herd immunity to work, we need over 95% of the population to be vaccinated. And then those remaining individuals that can't get vaccinated, maybe they're immunocompromised, maybe they are allergic to the vaccine itself, for whatever reason, maybe they can't be vaccinated, maybe they're too young to receive the vaccine. Those individuals can be protected by everyone around them that's immune to the disease. That's called herd immunity. Getting vaccinated serves the common good because it increases herd immunity, and it also serves your individual good because it makes you, or the individual getting vaccinated, much less likely to experience the full-fledged infection and all the symptoms of the infection. If you were born after the year 1970, you probably don't have a very robust memory of what it was like before vaccines existed because you've grown up in an era of vaccines. So it's tough to remember, it's tough to imagine what the side effects or the risks are of not getting vaccinated. Before the measles vaccine was made available, millions of people got measles every year in the United States, and hundreds of people died from measles every year in the United States, and over 1,000 people every year were permanently disabled from measles in the United States. Our childhood vaccines that we've developed as a society have saved approximately 2.5 million children every year worldwide from an early death. The risks from infectious diseases are almost always going to outweigh the chance of an adverse reaction to the vaccine. Now, there always are those few exceptions. If you know that you're allergic to eggs, you probably should not take a vaccine that has egg components in it. That you would be one of the exceptions to this bullet point. Or if you're someone who is immunocompromised, you absolutely do not want to take a live version of a pathogenic vaccine. So, who should get vaccinated and when? There should be a lot of caution exercised when we give vaccines to immunocompromised and pregnant patients. The recommendation currently is that vaccines should be given for all typical childhood diseases as soon as the vaccine is available and at the right time frames. Only in very specific circumstances should an adult receive a vaccine. Generally speaking, those are people that work in healthcare and are exposed to many pathogens, if you travel, or if you're in the military. A revised recommendation that's recently been released is that vaccinations in adults that are more aged or older can be given to boost the effectiveness of a vaccine. For example, a tetanus vaccine needs to receive a booster shot every 10 years to be effective. So generally speaking, what ends up happening is most people, when they step on the rusty nail, just get the booster shot right then and there. So, concept check, class. Determine which of the following statements below describes active or passive immunity. You can go ahead and rewind the video, flip back in your notes, or check your textbook for the answer. Pause it and get me an answer. 1, 2, 3, 4, 5. All right, let's look at infusion of gamma globulin. Is that active or passive? It is passive. Your body's not making the antibodies. If you recover from having influenza, you make your own antibodies. That's active. If you are a recipient, not a receipt, a recipient of the influenza vaccine, that is going to be active immunity. It's active artificial. So I'll put active artificial here. Recovery from influenza is active natural. Infusion of gamma globulin is passive artificial. And antibodies going from mom to baby through breastfeeding is passive natural. If you have any questions regarding vaccinations, please feel free to post them on the discussion board or shoot me an email. Happy studies.